We're up over 50 if you want to start.
Okay, thank you. Hi, everybody. My name is Dr. Cindy Malone. I'm from Cal State Northridge, and I run the CSUN UCLA Stem Cell Scientist Training Program here. Welcome to our After Lunch Club session. We are going to, this is session 2, our scientific session number 2, and it's covering autism. And we are going to, I'm going to give a little intro about our 3 speakers. And then after we watch and listen to their talks, we will have a little question and answer session. So I'll come back for that. But let's do a little intro. We have 3 distinguished speakers. We have Daniel Geschwin from UCLA. We have Joachim Hallmeier from Stanford University, and we also have Alison Motri from UC San Diego. So I'm going to give you a little brief about each of our speakers. They're total rock stars and have done it all. So I would essentially take up this entire time just reading their bios to you guys. So I'm going to give you some highlights about what these guys are up to and how great they are. We'll start with Dr. Geschwin. He is one of our CSUN UCLA program all-stars, and he's always game to take our Bridges students, which is great. And so we're hoping again for this fall, I'll be hitting him up with some CVs this afternoon. So be ready for that. Daniel Geschwin is the Gordon and Virginia McDonald Distinguished Professor of Human Genetics, Neurology, and Psychiatry at UCLA. He is Senior Associate Dean and Associate Vice Chancellor of Precision Health. And he has done it all, founded the UCLA Center for Autism Research and Treatment with Marion Sigmund back in 2003, and has been working in the field of autism for over 20 years. He has scientific service awards from Autism Speaks, gold medal awards from the Society for Biological Society, an elected member of the National Academy of Medicine, and again, one of our faves for mentorship of our Bridges program. And we also have next up, we have Joachim Hallmeier from Stanford. And again, he has done it all. He's the professor of psychiatry and behavioral sciences. He's been working in neuropsychiatric disorders for over 30 years and is involved in lots of genome-wide studies and linkage analysis as well. He has worked with a CIRM-funded grant in a large cohort of children for ASD, and he's established iPSC lineage lines in that disorder. So his work has been essential for autism, and he is a great asset and is going to give us a really great talk. And then my next little quick overview is Alison Motri, and again, he is from UC San Diego. He is a professor in pediatrics and cell and molecular medicine, and he's director of a stem cell program there at UC San Diego. Again, he's done it all. And he has been here, and he has his bachelor's degree in biological sciences from the State University of Campinas in '95, and a PhD in genetics in 2001 from University of Sao Paulo in Brazil. We're lucky to have him move up to the Salk Institute for postdoc, and now he is working in brain organoids and pluripotent stem cells in these autism spectrum disorders. And he is recently the prestigious NIH Director's New Innovator Award recipient, Young Investigator Awards, and the rock star in innovation. I told you guys they're rock stars, and he officially is getting one of those awards amongst many others. So with that, I would like to welcome them and welcome all of you to these amazing talks by our amazing mentors in our program. And I will be back for the Q&A session. Take it away.
Good morning. It's with great pleasure that I come to you today remotely discussing how we're working to develop valid stem cell models for neurodevelopmental disorders. What I'm going to tell you first is introduce a little bit about autism, which is the main neurodevelopmental disorder that we study. And then I'll talk to you about the challenges and strides that we've made in this area. These are my disclosures. I'm not going to be talking directly about any of the products or things that these companies are working on today. In 2008, there were really just a handful of genes, little understanding of mechanism, and major pharmaceutical companies were withdrawing from central nervous system diseases in general, and there was little interest in autism. But as genes began to be identified, it really was feeling like we were on the threshold of a new understanding. And indeed, 8 years later, by 2016, we were able to identify specific molecular pathways and circuits in autism. At this point, we had more than 200 candidate genes, much clearer mechanistic models, many advances, and major drug development efforts in autism ongoing. And this is from a review in 2016 showing several things. One is that there's a component of autism shown down here. At this point, about 10% of autism, but predicted to account for up to 20%, that is a fifth, of rare genetic disorders, some of which are inherited, but most of which occur de novo, that is like Down syndrome. They're genetic mutations that occur in the germ cell, and that therefore aren't inherited from the parent. Parent doesn't carry it. Or you have other syndromic, X-linked, etc. But one can see that there's a piece of autism, about 20%, that's predicted to be a collection of rare genetic disorders. And these can be studied because they're largely thought to be single gene mutations. And so what we know now is we predict, based on the discoveries that we've had so far, that there are likely to be many genes involved in autism, that none account for more than 1% or so of cases. That is, again, this notion of a collection of rare disorders. And there's strong pleiotropy in that many mutations, most of these mutations can cause many different kinds of clinical syndromes, ranging from autism, which involves the impairment of social cognition and the presence of repetitive restrictive behaviors, usually onset before age 3, and intellectual disability. And only about a third of cases of autism have intellectual disability, so they're not one in the same. You could also have intellectual disability with an epileptic encephalopathy due to some of these mutations. So again, strong pleiotropy. So the notion is these, in thinking about them and framing them, these aren't necessarily autism genes. They contribute to autism risk. Excuse me. But what they really do is disrupt brain development during critical periods, thus leading to susceptibility for a range of neurodevelopmental disorders. So the challenge is that advances in genetics and genomics have identified many genes, and these genes provide now targets for neurobiological exploration of mechanisms and therapeutic development. However, these findings highlight extreme genetic heterogeneity. So one of the questions is there going to be convergence? Are there going to be groupings of these mutations that fall into specific biological pathways, developmental stages, or brain circuitry? And so we've been asking that kind of question. So we asked early on in 2013, when the first tranche of genes was identified in this paper, Parikshak et al. And Cell, and then much more recently in Walker et al. And Cell, both who were graduate students in my lab. We asked when and where do autism genes act, and we were able to show they act early in development at the peak of cortical neurogenesis, prenatally. That doesn't mean all genes act there, but when we asked, are they enriched in a particular process? This is where it was. And then when we asked, is there any biological process? They were enriched in early transcriptional regulation, again, peaking during cortical neurogenesis. And so these large effect mutations that I showed you mostly involve transcriptional regulation, and then the smaller effect size mutations mostly involve synaptic development. And we could see when we looked and asked when and where these genes are expressed, and this has been borne out in subsequent work, that they're expressed in the superficial layers of the cortex. And that's important because those layers connect regions to each other. They're the regions that connect different parts of the cerebral cortex, including the two hemispheres, to each other. So this fits with about a decade of research in neuroimaging that's shown long-range cortical disconnectivity in autism. So now we're seeing a merging and of the kind of genetic data and the neuroimaging and cognitive data. But if we back up a little bit, we now have to ask, how do we develop therapeutics for this complex disorder? What do we use? And so, of course, we benefit from the enormous strides forward in the stem cell field, driven by the findings of initial Yamanaka. This was one of the first papers we were able to collaborate with Ricardo Dolmetsch's lab when Sergio Pasca was a postdoc there to identify, using iPSC-derived neurons to begin to study the mechanisms of one of the rare forms of autism, Timothy syndrome. But again, the point that Ricardo Dolmetsch and I made in a review prior to this was there was the promise of having human brain in a dish at this time, but it was a lot of promise without a lot of clear validation. And again, one has to be extremely careful when making claims about these models. Do we have evidence? What do they actually model in vitro? And so as we began to look at the time two-dimensional models, these 2D models of cortical development in the dish, we looked at both human neural stem cells that are primary cells and iPS-derived neurons from several of our collaborators, and used genomic tools, which can give you an unbiased view. So you can ask the question, how well do my stem cell models in vitro model development, and what is modeled? And we developed a number of tools, a transition mapping tool to look at what transitions are occurring in vitro, a machine learning tool that determines the developmental maturity and regional identity. And so what we showed in there is we have the beginning of tools to begin to ask, what are we modeling and when are we modeling? And in this showed, using this quantitative framework, that the development of the 2D models really didn't get past mid-gestation. So you could model anything up to about 24 weeks in vitro, but past that, you weren't really seeing very much past that. So you certainly not birth or post-birth. As we began to work with Sergio Pasca's lab to apply these methods to the more advanced three-dimensional cultures, we saw that they were able to advance developmentally more than the two-dimensional cultures, which was really important. They also contained, in kind of very similar patterns of expression, the gene expression of a lot of the autism mutations that are just shown to the right in the midnight blue module. So more recently and over time, this took many years with Sergio Pasca's lab, we've asked how well do 3D cultures model prenatal and postnatal development. We used multiple genomic techniques looking at RNA and transcription, DNA methylation, RNA editing, and also John Huguenard and Sergio looked at physiologic switches such as NMDA receptor switching that is known to occur around birth. We have high reproducibility of these cultures over time, greater than 0.9. And what we show here, using the transition mapping tool that I mentioned to you before, prenatal stages go up to about 7 in vivo. These are using the brainspan data, and 8 is postnatal. And one can see that between 250 and 300 days, these cultures begin to resemble and have components that are resembling postnatal stages. So indeed, as these grow, they are recapitulating human development. In fact, it looks like there's almost an intrinsic clock that is being measured. Here we're looking at DNA methylation age on the y-axis and differentiation day on the x-axis, and we can see this almost linear relationship going on, suggesting the preservation in vitro of this in vivo clock. We also see that autism risk genes aren't monotonic. They fall into different trajectories or clusters, some that are very, very early and more broad, some that are at mid-gestation, so to speak, and some that come on a little bit later and are actually enriched in astrocytes, which is consistent with astrocytes coming up at about 150 days in culture. And again, we can see that these clusters also correspond to distinct functional groupings as well. So we have the early chromatin modifiers and histone modification that regulates gene expression during cortical neurogenesis, later synaptic signaling, and forebrain development and regulation of behavior. We've created a browser that allows you to look for any genes that you're interested in neural development and see what their trajectory looks like and if they're expressed in these cortical organoids. So knowing this, we've been working in a CIRM-funded project to now ask about some of these rare mutations and ask, do they converge? Do they have different properties, et cetera? And the 9 different, we're looking at 8 plus idiopathic autism versus controls. And we have about 10 controls, about 10 idiopathic autism, and then various numbers of these others. We differentiate for 100 days only at this stage because we're looking for early patterns of overlap. And we checked gene expression at 25, 50, 75, and 100 days. And this is the number of individuals, the number of lines, and the number of differentiations. This is really an unprecedented study in its scope. And what we're able to see, and just summarizing years of work into one slide, is a really interesting clustering, such that, for example, SHANK3 mutation clusters with 22q13.3 deletion, and this is kind of expected and a kind of validation because the SHANK3 mutation is within this deletion and is thought to be the gene that causes the symptoms here. That's the causal gene. And so we're able to see, using multiple clustering methods, many different ways of looking at it. We've kicked the tires on this over and over again, and this is a very robust clustering pattern, such that certain disorders actually cluster together. And we can see overlapping patterns of dysregulated gene expression in some of them. Now, because we had a large number of the 22q11.2 deletion, Sergio's lab and John Huguenard were able to do extensive physiologic investigation. And this was recently published in Nature Medicine. And it just, this is work that actually Sergio started when he was a postdoc in Ricardo's lab. And it took nearly 6 years from beginning to end. So we did gene expression study, and we show that there's a calcium defect in calcium signaling. This is validated physiologically, and actually Sergio's lab showed that the gene DGCR8, which is within the 22q deletion, when knocked out, recapitulates this phenotype shown here in yellow, and that they were able to find a drug, raclopride, that was able to rescue this, shown here again in yellow, which is really quite remarkable and shows the power of using these models to identify physiologic deficits in drugs. Of course, there's a little bit of, there's a lot of translation that needs to be done between this in vitro work and doing something like this in humans, but it's a great start. We've also been working, not just, if we have to do this kind of painstaking physiologic work for every mutation, it's going to take a long time. We've been asking if we could use gene expression profiles to find drugs. And this is facilitated by connectivity maps, that is, maps of what happens to cells after treatment with known compounds. So there are thousands of expression profiles now in databases, and we can map gene expression in tissue models or patient tissue to these drug profiles to find a drug that can match that pattern or reverse that pattern and then treat it. The 2 papers shown below, Swarup et al. And Retzak et al. Are actually examples where we use this in neurodegenerative disease to identify drugs that will protect neurons from neurodegeneration. Those are 2 very powerful proof of principles. We have not yet done this in autism, but are moving forward in that direction. And I'm quite optimistic, although again, cautiously. So what I've told you is that autism has a large genetic component. We've found dozens and dozens, over 100 ASD risk genes. They converge to impact early fetal brain development and the development of the cerebral cortex. That doesn't mean they're exclusively expressed there, nor does it mean that's the only process going on, but that's one major process that's being developed. And fortunately, we've shown that this period can be modeled with high fidelity in human iPSC-derived models, especially 3D cultures. And this is very important. We've used unbiased genome-wide functional genomic methods to show this. And it provides a foundation for understanding disease mechanisms and drug discovery. Well, I have to thank, obviously, this work was done by a lot of folks in my lab, shown here several years ago. I've starred many of the people who have left the lab who have contributed to our work in autism, but this work really that I've talked to you has really been driven by a collaboration with Sergio Pasca's lab at Stanford, who is an extraordinary pioneer in this field, as well as my other collaborators at UCLA, Steve Horvath, Mike Gandol, Catherine Plath. Some of the work that's unpublished was done by Katie Eyring. Aaron Gordon has led these CIRM studies and these cross-disorder iPSC studies, as well as the maturation studies. He's a very, very talented senior postdoc in the laboratory. Again, we thank our funding sources shown here, as well as our large collaborative networks such as PsychEncode and BrainSpan. Thank you very much for your attention.
Today, I want to talk to you about studying autism spectrum disorder using iPSC models. And I want to give you my view, what I see basically as the challenges and what I see as a big chance in what this technology offers. Now, before we go into the iPSCs, what are autism spectrum disorder? Autism spectrum disorder is a term basically which was created to bring together different subtypes of what was previously called pervasive developmental disorders. These were autistic disorder, Asperger's disorder, and pervasive developmental disorder, not otherwise specified. All these was basically now subsumed under this one term, autism spectrum disorder. It's important, it's not disorders, it's disorder. So it's one single disorder. Now, why did this happen? The reason for it is that the core symptoms were really shared basically amongst these subtypes. But these core symptoms were basically associated with a wide range of outcomes and etiologies. And any attempt basically to subgroup these have failed basically, despite numerous attempts by scientists, including myself, to somehow create a reasonably good or clinically valid subgroup, it entirely failed. So what are these core symptoms? These core symptoms occur basically in two domains. The one is social communication and the other one is repetitive behaviors. In the social communication areas, there is a lack of social emotional reciprocity. There's a deficit in nonverbal communicative behaviors. And there's an impairment in developing and maintaining and understanding relationships. On the more repetitive behavior areas, what we find there is often very stereotyped and repetitive motor movements, an insistence on sameness, highly restricted and fixated interest, and hyper and hyporeactivity to sensory input. It's important to note that these symptoms must occur in childhood and they must impact daily functioning. Now, from a less medical model, autism spectrum disorders has also been characterized as a combination of strengths and weaknesses, where the weaknesses are really in the social domain, in the communication domain, in the social interaction, and the repetitive behaviors. It's important to note here that about 10 to 20% of children with autism do not develop language. So the strengths are then that many of these children are detail-oriented, have highly developed visual skills, and are extremely persistent. Now, on a population level, what we find basically that roughly about 1 out of 64 children is currently in the US is diagnosed with an ASD. There are significantly more males diagnosed with ASD versus females. In about 30 to 70%, the children with ASD score in an IQ range of less than 70. And a large percentage is estimated between 10 to 30% develop epilepsy. What is the etiology? Unfortunately, we don't know the cause of autism. We don't know one cause of autism. I can tell you this was causes autism. What we know is that it is a combination of genetic and non-genetic or environmental influences. And I want to talk a little bit about the genetic causes because they play a big role in the iPSC studies. What we find basically in children, and there are some very large studies have been carried out meanwhile from these tens of thousands of individuals with ASD. What's basically found is a higher rate of rare variants, which are found in about 10 to 30% of cases with ASD. These rare variants increase the risk for a child to develop ASD significantly. However, none of this mutation is associated with more than 1% of these cases. And the 1% is really it's a high estimate. It's much, much lower for most of them. So what about the rest, the 70 to 90% of the cases? There are common variants which play also a role. Several of these common variants have been found, and we will know many, many more in the near future. However, the conferred risk increase is low. It's much lower often than in the rare variants. The rare variants are basically rare, and the increase is risk much more dramatically than the common variants. However, none of these variants by itself is sufficient for a child to develop autism. There must be other factors, or what also likely is that several of these genetic factors come together. So the question is basically, how can we use now this genetic information where we know now a lot of variants which basically increase the risk of autism? How can we translate this? How can we find out what is the mechanism? How we move forward? It's clear the missing link is that we would like to look at how the brain of these children develops. Now, this is very difficult for obvious reasons, because contrary to, let's say, cancer, where we can have a sample and then we can study it, we cannot have a brain sample. So basically, the missing link is really how we get access to neuronal tissue. And that's where the iPSCs come in, because as you know, with an iPSC, we reprogram the cells and then we can grow whatever tissue we want. And we are able basically then to grow neurons. So what we get basically are an unlimited supply of neurons from the individual with the disorder. I take a cell from the individual with ASD, transform it, and then I get my neurons. So what's preserved there is a genetic background, but what's basically forgotten, if you want, or erased, or however you want to call it, is the environmental factors. So basically, we can then compare the development of the neurons from individuals with the disorder, in this case with autism spectrum disorder, to control neurons. And we know that these neurons, the development of these iPSC-derived neurons is pretty close or pretty similar to the development in humans. We can also compare then the functioning of these neurons and the organoids from individuals with ASD to control neurons. These organoids, basically, you let them, what you do is you let them grow, and then they basically build synapses, they connect to each other. So it's like a little it's like a little mini brain now, it's sometimes called. So we can also, and that has been done very limited in autism, we can also manipulate the development of function of these neurons. So basically, if we would know, or once we know more about the environmental factors, we can start to model these. For example, if we think that really, let's say, toxic pollutants play a role, we can expose these neurons to these pollutants and then figure out basically what's going to happen. And also ask the question whether individuals with the disorders react differently. However, we must be very aware that these neurons provide only models. They do not fully recapitulate the molecular and functional signatures of their in vivo counterparts. They are not like a neuron grown basically in utero. So when we started out, and this is long ago, the first question basically we asked is, when we heard about this technology of iPSC and started to use them, the first question basically is, can we even find something new? Basically what we started out to do is a proof of principle. Can we find the effect of the mutation? Is it basically represented in the functional property of the neurons derived from a skin cell? At this time, all the iPSCs were derived from skin cells that we studied. Now they also derive from blood cells, so it's actually more common meanwhile. So what we did is basically that we put together a cohort of individuals who had these rare deletions which were associated with autism, which I said before, which increase the risk dramatically. And then basically we wanted to see whether we can find differences in neurons from individuals with these genetic mutations compared to control neurons. This was before you could do this with CRISPR, okay? I want to just show you very briefly something recently we did was where we studied one in a little bit more detail, which is 22q11 deletion syndrome, which is associated with autism, but also with another psychiatric disorder, schizophrenia. We studied them basically, in this time, we let them grow to this little organoids or mini brains, and then basically we studied the neuronal excitability. And then we basically, what we next did is also that we influence this excitability with different compounds, which we know affects calcium. And also we studied one very specific gene, DGCR8, which is in the deletion, which is basically deleted in these individuals. And what we basically found were changes in spontaneous firing and depolarization induced calcium signaling. We found an increase in excitability and impaired depolarizations of voltage-gated calcium channels. We found abnormalities in the resting membrane potentials, which could be rescued with antipsychotic medication. And we could also show that the heterozygous loss of this gene DGCR8 was sufficient to phenocopy the functional defect. So one of the hypotheses would be that what we see in this syndrome is due to the loss of this gene. So basically what we showed with this, and we showed this in several of these other disorders, and as a colleagues, I mean, there are many other people in our group which are working on those. So we know now that we can definitely find the effect of the mutation in the functional properties of the neurons derived from the skin cells. Now, big question is, do these different disorders we have studied, do they have something in common? And there are some emerging themes. There's not a neurophenotype where we can put our hand on and can say, okay, that's now characteristic for these disorders. Well, that's where they converge. But we found some emergent themes which really point towards the synapses and to migration deficits. Interestingly, they are really converging with what you know about the genetics generally. So what about these 70 to 90% of ASD cases without the mutation? There are several studies which have been typed. This is not complete. These are just some of the more prominent ones. And the issue with most of these studies is that the numbers are small, and any conclusions to be drawn are premature, I think, at this stage. What we need is a confirmation in much larger numbers of lines, and we need also to confirm it in using different methodologies. One of the biggest challenges is then to link this basically back to the clinical phenotype. Basically, what we need, I think, is a much, much bigger boat. If we want to really study autism on a more general level, I think we need a big, very, very, very big boat. Now, what's the problems? What are the challenges? The biggest hurdles are costs, because this methodology is labor intensive. It's availability of samples. And it's in-depth phenotyping. It's basically we really have to understand what are the clinical symptoms of these individuals. We started basically we got funding from the CIRM here to develop a repository of a larger group of individuals with ASD with no known mutation. And we are studying, currently we are starting to study, unfortunately I cannot show results at this stage yet. Our goal is to study about 100 cases and 100 controls. We have a total of 164 subject iPSCs now, from which are all diagnosed with the gold standard methods. And we have a control group of about 134 typical development group. What we are also doing is that we develop functional assays, which we hope are very robust and that we can repeat them. We try to automate this as much as we can. So, what do we know so far? To summarize a very complex field, actually, when ASD is no mutation. Again, there is no clear-cut neuronal phenotype. However, what's clear is that even in relatively smaller samples, and even in our preliminary results, what we can find is that there are changes in the neurons compared to the control neurons. there are some emerging themes, and I want to call them themes, definitely not phenotypes. Cell proliferation and neurotransmission is what comes out there. So what are the challenges, to summarize here? The biggest challenge will be to relate basically whatever we find then to the clinical symptoms and to the behavior. The heterogeneity of the disorder presents a huge problem. It's very unlikely that there will be only one phenotype. There will be basically multiple phenotypes. As I told you already, the effect size of the known genes are low and unspecific. There's not a single gene which is really where you can say this gene causes autism, or with a mutation like this, all the children have autism. Sample sizes are a huge problem. Replicability of the methodology, and you hear more about it, the other talks about it. And I think another problem is also a selection of the neural phenotypes. There are a lot of technologies available, but just we can do it technically doesn't mean it makes always sense to do it. So how to select these, and how can we as a group basically agree that these are probably the most promising one, though we can do it in different labs and replicate each other. And with this, I want to thank all my collaborators, which are numerous, not all of them on there. All my studies are very, very collaborative, and I'm doing only a very little small part, mainly on the clinical side. And also my funders, CIRM, the Simons Foundation, and the NIMH. And with this, I thank you for your attention.
Thank you so much for the invitation to talk and share my research with you guys. It would be a real pleasure. So my lab focuses on modeling autism using brain organoids. The reason why we use brain organoids because autism having like a strong genetic factor actually starts in uterus, and it's experimentally difficult to access the human brain inside the uterus. So we rely on models that can actually mimic or model neurodevelopment outside the human uterus. So that's why we turn into brain organoids. And brain organoids was first described in 2008. That's exactly when I started my lab by Yoshiki Sasai, who showed us that you can actually grow human pluripotent stem cells and neuralize in 3-dimension floating spheres that will self-organize and form tissues, neural tissues that are similar in anatomic organization as the human fetal brain. I was very excited with this research, and that's why I follow on modeling neurological conditions with this protocol. But of course, I mean, brain organoids are not brains. There's different protocols out there, but all of them have intrinsic limitations. These are intrinsic limitations or shortcomings from the protocol itself. For example, most of them don't have strong neuronal activity. These neurons are quite immature. These organoids are not vascularized. We are missing cell types. Even the culture condition is not ideal. My lab actually decided to create a new protocol where we optimize the condition for human neurons, so human neurons can fire spontaneously. And we start from single cells, and we end up with spheres that contains 2.5 million neurons. And the protocol is very simple, very robust, requires only 3 steps. So this is Isabel, a postdoc in the lab, and she's just finished a batch of human brain organoids. So you can see all these white dots. Each one of them is one of these cortical brain organoids. So you can actually visualize them, and you can see that in a single batch, you can actually have thousands of brain organoids for different experiments. So if you, this is a model of a brain organoid in representation in 2D. So you can see that on top is where the core of the organoid relies, and you can see the migration of the neuroprogenitor cells following the cables or the radioglia cells, allowing them to migrate from inside out, exactly how the human brain develops. And as they migrate to the external part of the organoid, they stop and start to differentiate and form connections. So if you cross-section one of these organoids, you actually have a ventricular zone surrounded by neuroprogenitor cells. And again, in white here, you have the radioglia cells. These progenitor cells will migrate and form a nice cortical plate. So you might ask, what kind of cells you have in there? And the diversity of cells or populations is quite dynamic. It really depends on where you are in the protocol. As I mentioned, we start with single cells and we end up with 2.5 million neurons in there. So this is a snapshot at 4 months. You can see that most of the neurons are glutamatergic excitatory neurons. At that stage, you already can see two populations. These are upper and lower cortical neurons. The population of progenitor cells continue to differentiate. At this stage, they are becoming more and more gliogenic. So you see the migration or the differentiation in astrocytes, no longer neurons. And astrocytes start very discrete, like 5%, and they end up being 50% of the population. So 1:1 ratio to neurons. And at that stage, we also see the emergency of GABAergic neurons. They will populate the organoids in about 15–20%, and there are 3 major classes of inhibitory neurons that we can detect by single-cell sequencing. So this is, there are also possibilities of adding cell types that coming from different embryonic origins, such as microglia. We recently developed a protocol to infuse microglia in these organoids, and we can keep them for long periods of time. So this is an organoid where the microglia is here labeled in red. You can see we can keep them for 18 months and counting. So some of these overcomes, as you can see, will actually have new findings in 5 years from now. Others are more difficult. But how much of these organoids actually represent the human brain? And most people attack that question with transcriptional profiling. So do we. We use data sets such as the BrainSpan and tools such as Context to compare the global gene expression of organoids with human brain neurons or human brain regions. And as the organoids progress and become more and more mature, you can get to late fetal, even postnatal brain, more or less a brain that it matches like 6 months to 1 year old human brain based on this kind of analysis. But really, we are interested in networks. Can this new protocol actually provide neurons that forms, organize, in complex networks such as we expect for the human cortex? That would be crucial if you want to study autism. So the reason why we were doing that, it is because we want to have an idea of the large scale of brain signals. So these are 2 examples where if you just do a patch clamp in a single neuron, and I'm taking the analogy here of a stadium with the brain organoid. If you just look at some people here, individuals, you don't understand what's going on in general with the entire population. It's the same for organoid. If you just patch a single neuron or a couple of neurons, you might get the activity of those neurons, but you have no idea how the networks behave. That's why we turn into multi-electrode arrays. So these are electrodes that are printed in the bottom of a dish. You can place the organoid on top. And the beauty of that is that it retains the 3-dimensional structure of the organoid while you can record longitudinally. So you can see that we can generate these raster plots here where you have the different spikes that is captured from these electrodes. So you can actually understand how the networks evolve over time. And I was not very optimistic about that because most of the previous studies using 2D human neurons were actually measuring activity that are below 5 Hertz. And we want something that is close to the human primate cortex, that it would be something about 20 Hertz. So these are overall brain activity. So we always thought that the 2D cultures, because they peel off, they cannot be kept for long periods of time. That's why they never reach that level of maturation. But others have tried to place organoids on multi-electrode arrays as well. So this is the work from my colleague, Paola Lotta, where she played 32 weeks old organoids on multi-electrode arrays and also could not cross this 5 Hertz threshold. So fortunately, I have a very tenacious postdoc who decided to do that. And using this new protocol, you can see that we start observing like a large increase of synaptogenesis when the organoids are about 25 weeks. That's when the astrocytes start to appear in the organoids. And then you see almost exponentially an increase in electrical activity, reaching like something close to 20 Hertz when they are about 40 weeks or 9 months of age. We can measure activity from excitatory neurons as well as inhibitory neurons, doing some pharmacology. So we confirmed that we have all the units in the same organoid. So we don't need to do different types of organoids to have both excitatory and inhibitory neurons. And we also confirmed that the neurotransmitters are being released by those neurons. So it's not neuronal markers or gene expression, but we can actually see the release of these neurotransmitters by mass spec. So now that we have that, and because there is so much activity, we wonder if we are actually seeing neural oscillations. And neural oscillations are these activity that you can record by placing the electrodes throughout the skull of the human brain. So this is what we call an EEG or electroencephalogram. So that would be important because it would allow us to cross or to bridge all the basic biology that we can get with these organoids to the human brain or behavior or cognition. This is something that cognitive neurosciences have been doing by correlating these neural oscillations with human behavior. But there is a huge gap with the biology. So maybe we can give like one step further to bridge that gap using brain organoids. So that was the motivation to look for these neural oscillations. So we use the data from the multi-electrode array. We create this spike raster, and then we combine in population spiking, and from that we generate the local field potential, which is really the basis for these neural oscillations. And in summary, what we have is that we don't see the formation of neural oscillations by 2 months. We see the spontaneous emergence of some of these oscillations starting at 4 months. By 6 months, they're very clear, but still very stereotyped. And by 8 months, they become highly complex. So this evolution of these networks is exactly what you'd expect for a complex network such as the human cortex. So we're quite happy with that. But the question was, how similar are those with the human brain? So we decided to attack that by creating a machine learning algorithm where we train the machine using EEGs from newborn prenatal baby brains that were born between 25 and 38 weeks. So we train the machine gets really good. It can tell the age of the subject by just looking at 2 or 3 EEG features. Once we train the machine, we start feeding the machine with the data from the organoid. So this is totally unbiased. The machine has no idea if the data that it's coming is from the human brain or from an organoid. And then we ask, can the machine matches the age of the organoid based on the training from the human brain? And the data, it's beautiful. The machine gets totally confused, cannot distinguish an organoid from the human brain anymore. So here is I'm plotting the predicted age versus the actual age here. And the perfect prediction would be in this dotted line. The human brain is the black dots, and the organoids, each organoid is one of these blue dots here. So you can see the variability of the organoid is definitely higher, but it follows the same trajectory as the human neurodevelopment. So that was quite amazing. So it shows that that very simplistic and reductionist model can actually mimic a complex activity such as the formation of cortical networks in vitro, something that, of course, was never reported before. And I like this article from the New York Times talking about our research because it raises also this ethical implication. As scientists build better and better brain organoid models, we get closer and closer of the human brain, including in electrical activity. And we should not forget about the ethical implications of this research. So we are trying to mature these networks even further. And we do that by input and output. For example, after 40 weeks or 9 months, we are starting to add a visual stimulus in these organoids by plugging them with a retina organoid and a thalamic organoid. So you can see the photoreceptors with specific reporters and the projections of these photoreceptors into the cortex. The hope is that to mature the photoreceptors so we can stimulate with light while record from the cortex. We also do that in a very artificial way. This is our collaboration with CENI here at UCSD. It's a project sponsored by Microsoft where we are stimulating these organoids using a robotic interface platform. So what you see here is a robot that is learning how to walk. based on the electrical activity that's coming from one of the organoids inside our incubator. The activity is sent to the robot wireless. So we are now adding sensory information. For example, these two eyes, these are not eyes, these are infrared sensor in the robots that actually tells when the robot is approaching an obstacle and stimulates the organoid using optogenetics. So the network gets stimulated, the computer notices that there is a new stimulus in there and send a second command to the robot, such as walk back. So our hope is to see if we can induce plasticity in these organoids and actually learn how the human brain learns in this very innate system. So it's very artificial, but nonetheless, we hope that this will actually lead us to different oscillatory behaviors in the network. So the other system is by growing organoid at the space station. We send organoids to grow there in microgravity, hoping we can mimic what happens to the astronaut cells. This is inspired by the NASA Twins experiment, where long-term exposure to microgravity actually shows some signs of early senescence. So we actually want to use the organoid to age the cells or the neurons, so we can model a later onset condition, or even push our organoid to a more mature stage. So we do that by collaborating with bioengineers to create boxes like this one that is fully autonomous to grow brain organoids. And really the only thing that the astronaut has to do is to plug these boxes in one of these cabinets in the background. So give us access to what's happening there, so we can control the temperature and we can visualize the organoids. And the idea is to bring them back and start doing some analysis to see if they actually senesce in those conditions. So going back to autism, can the model that we currently have be used to treat autism? So my lab creates a series of isogenic and patient-derived cells, so we can create organoids from all these conditions to test pharmacological drugs, as well as gene therapy and gene replacement. The hope is that we can speed up this preclinical experiments and move into clinical trials. And I will give you one example of a very successful condition. This is modeling Pitt-Hopkins syndrome that's caused by a deficiency in a gene called TCF4, transcription factor. So it's a very severe neurological condition, more towards the end of the spectrum, where it's more severe. So you see absence of speech, severe constipation. There are generalized hypotonia, so most of these kids cannot even walk. So very severe. Can we do something about it? Unfortunately, TCF4 has several isoforms, and we didn't know which isoform was really expressed in the brain. So we use our organoids to learn that isoform B is actually the one that's driving the expression in brain cells. And we create a vector by placing the TCF4B isoform downstream to a minimal promoter that is followed by these micro E5 binding motifs of TCF4. So we can actually place different sizes of these binding motifs to create a feedback negative loop. So there's never overexpression in the cells. And we packed all these into AAVs, and we used to infect the organoids. So the organoids from Pitt-Hopkins conditions don't express too much of the TCF4. Most of the genes are loss of function, and they have like severe abnormalities. For example, the number of neurorosettes also is affected. But we place those on multi-electrode arrays, and we see that there is a correction or an increase in electrical activity, rescuing at least partially the networks from Pitt-Hopkins condition. Even more impressive is the fact that these AAVs can actually correct the structural defects of these organoids. In the patient cells, we always see these organoids that have these neuronal scars or lack of neuronal rosettes. And by adding the AAVs early on in the process, you can actually rescue that. That was very interesting to see. So I will stop here and list my take-home messages. We think that you can adapt to different protocols for different questions. Brain organoids mimics these very early stages of neurodevelopment, but there are limitations that we are working on it. And finally, we think that it can be a useful model for autism in testing potential therapeutic opportunities. Full disclosure, I'm a co-founder of Tismo. It's a company that uses brain organoids for autism. And I would like to thank CIRM and IMH as funding agencies and several of my lab members who participate on this research. Thank you very much.
Good morning.
Okay, hi everyone. Thank you so much. Those were some great talks. We have some time for questions. Of course, I did talk too long at the beginning, but okay, let's see. I will go into our question and answer. Our first question, since epigenetic memory is better preserved using a direct differentiation protocol, have there been studies to characterize and study trans-differentiated, as opposed to iPSC, ASD-derived neurons? If so, what have these studies shown? I'm not sure which of you this is directed to.
I can try to answer.
Okay.
Yeah, I mean, I think Mitra is correct. The transdifferentiation would preserve some of these epigenetic memories. For autism, I would like to highlight the work from Rusty Gage, my colleague here at Salk. He did compare some of the properties of autistic IPS-derived neurons and transdifferentiated neurons, showing that they kind of skip some of the developmental stages if you go towards directed differentiation. So it really depends on what you're trying to answer. What is your question? Maybe transdifferentiation would be good to mimic a more mature state. But if you want to recapitulate neurodevelopment in a dish, I think we have to go through the traditional steps.
I would also add, yeah, that's exactly right. Thanks, Alice. Yeah, I was just going to add on top of that, is that most of the time we're also studying large effect genetic mutations. And unless one is postulating that there's some epigenetic state being carried, which we don't have strong evidence for, there could be in some cases, but what we're studying are highly penetrant mutations, some less penetrant, right? And there may be, but, and so, for that, we're not as concerned about the epigenetic reprogramming, although it is an open question and a very good question.
All right, thank you so much. We have a couple more minutes if anybody wants to go in the question and answers and post. Otherwise, I'll ask a quick question while we're waiting. This would go to Alison. In the last talk, you were talking about using the micro E5 binding sites to drive overexpression. Do you guys know what transcription factor might bind there, or is it sort of still unknown or novel?
Yeah, I mean, definitely the TCF4, the transcription factor binds in there and self-regulates. So that was the rationale to use it. I'm not claiming that it's specific to TCF4, and I don't think anybody has explored that in human neurons. We are doing that right now. But nonetheless, at least in our system, it seems to self-regulate. So it's kind of a fail-safe gene therapy approach.
That looks really good. Okay. All right, here comes another question. Uh-oh, it's long. It's from Kelly Shepard. Over the years, various factors were proposed as contributing to autism: parental age, exposure to pesticides, etc. Has anyone looked at brain organoid models created from aged iPSCs or chemically treated ones to look for those environmental factors? Like we talked a lot about the genetic factors. So how about environmental?
I can start at that. Let's just say one thing. When one's making a model, there are different aspects of validity. And one aspect of the validity has to do with how strong is the evidence that the factor causes the disorder. And certainly paternal age is a strong risk factor for de novo mutations that are, that occur with autism. The exact mechanism for that is not well known, although it's thought to be related to DNA repair issues that change with age. there's no evidence that strong evidence that causally relates exposure to pesticides to, for example, autism, and the same is true for mercury and other things, although those things certainly can't be good for brain development. And so it wouldn't surprise me that high levels of any of those things can lead to a variety of terrible, unwanted neurodevelopmental outcomes. I think the key is to really, there's some large epi studies going on now, especially in Scandinavian countries where these things can be well controlled, where there's kind of public, large public health systems. And there they can really assess these perinatal environmental factors a little bit easier than we can here, where there's so many confounding factors and huge disparities in socioeconomic class and other things that can really, drive, some of these observations. So, so there are a few environmental things that have been seen a few times. There's some, the pollution study was pretty good. There are a bunch of pretty good studies, but they need to be, really validated and better understood, to really see what's going on. We know that perinatal factors, things that cause intellectual disability, or even things that increase risk for cerebral palsy, like anoxia, like severe viral infections that lead to high fevers, can increase autism. So there is a role for environment. It's just a question of exactly what and how specific these things are, rather than just bathing the brain in a bunch of bad chemicals that are going to cause poor neurodevelopment almost no matter what happens. I mean, there's no question we want to avoid things like that, but there's a big gap in the kind of causal connection between environmental factors and autism at this point, for the most part. But I think that these iPSCs provide a way to test that for sure. I think it would be very, yeah.
Sorry, may I just add something? The important point, I think, is that that the iPSCs are good to study the gene-environment interaction. I must admit, if we only want to find out how a neuron reacts to, let's say, a pesticide, I think iPSC is really an overkill in a certain way. There are lots of other models we can test it. What you can test with the iPSC is the gene environment in the model, and to put a little bit to confirm what Dan said. Unfortunately, we don't know any gene-environment interaction. No factor basically we could test. If we would know of a factor which specifically interacts with the genetics, then we are in the game. That's what we can test in iPSC models.
Okay, thank you guys. That's great. We're a little bit over time for break, so if you need to go have your break before our 2 p.m. advocacy panel, please feel free. If you want to stay, listen to some more, at least one more quick question. Oh, there's 2 more in here. That's great too. So if you need to go take your break before 2, go ahead. Otherwise, we'll cover a couple more questions. This one goes to Alison again, about your machines and how they're controlled by the brain organoids. If you can quickly, maybe briefly go over how I think the organoid gives information to the machine.
Right, yeah,
and
so the
vice
organoid
versa.
has no intention of controlling a machine. In fact, it doesn't even know that it's doing that. So
Right.
It's totally separated from the machine. We capture the activity, we process that information in creating these neural oscillations. Then there is a computer who actually tells which oscillation controls which leg of the robot, things like that. And that's how we have the robot moving. And then upon stimulus, which is the infrared sensor, we just stimulate back those networks. That changes a little bit how the networks were behaving, and the computer sees that and send a second command. So the ultimate goal is really to eliminate the computer from the middle and having the organoid directly in the robot with no interference, but we're not quite there yet.
Thank you, that's great. And then the last one, well, maybe not the last one, right now in the Q&A is, I guess this would be towards any the 3 of you would like to comment on pros and cons of 3D cultures versus organoids. So as far as modeling, scalability.
Maybe somebody should explain that a little further. Some people might use the 3D cultures and organoids together. There are ways of making assembloids where you differentiate, you'd make neurons, you make glia, even microglia, and then you assemble those together. And again, at that point, I think Allison made this point beautifully, you might miss the kind of key developmental epochs that are going on. One of the things that is also clear that I think is really fascinating and unexpected is how well how there's almost this intrinsic kind of process going on that is captured in vitro that parallels what's going on in vivo. So I think that further amplifies that if you're interested in that developmental process, you can't really hurry it. You have to watch it because that's what's being disrupted in a way. I don't know if that answers the question. Allison would be the resident expert on all the different methods probably.
Okay, sorry, Allison, I just got the nudge that we need to go because
Okay.
Greg has to put on the next one. I guess we can't be on while they're on. But yeah, that's an excellent question. And I'm sure the speakers will be around for any of the participants to chat with in some of our other forums. So for now we have to go. We have to let Greg do his job for the next session and take our break. Thank you so much. I really appreciate it. Good job, guys.
Thanks.
Thank you. Bye-bye.
Guys.
Bye.